All right, in 341, we're going to learn about how pedigrees can be used to determine how um, traits are inherited and determine who might be getting the trait. So this is since sickle cell is a genetic disease, this is actually very important for people who might think sickle cell runs in their family and the probability of them passing on to their child. So right now, I'm going to give a brief overview, and then you can take the pre-test. If you do a pre-test, you can do well on the pre-test. You can skip the pedigree portion and go right to the assignment. If not, then you'll have to do the pedigree portion and then do the assignment. So here's just your key vocab terms. You got genotype, phenotype, dominant recessive trait. Again, you should remember those, but I'll go over them real quick. So you have two alleles. Allele is a type of a gene. So if we're talking about purple, we got the gene for the purple flower, and the recessive is the weaker one. So they have to, if you have two recessive genes, you'll have white. But if you have one recessive gene, the dominant overrides it. So you'll get a purple flower. All right, so dominant is always expressed when it's present because it's the stronger, it's the bigger letter. All right, so those are the two. The two alleles would be recessive for white, dominant for purple, and then the dominant overrides the recessive allele. Then we have those terms for homozygous and heterozygous. So a homozygous genotype can be either homozygous dominant, meaning you have both of the same alleles, and it's both a dominant, so this would create a dominant trait or dominant phenotype, or you could have homozygous recessive, and this is for a recessive trait. This is the only way you can get a recessive trait. Whereas we have carriers or heterozygous, hetero meaning different, homo means the same. This has one of each. Now, we never really write it like this. It's usually always like that one. But they're both heterozygous genotypes. It's just the order of their going. And these are carriers. It means they have one dominant and one recessive allele. And this usually always gives the dominant. These guys are always dominant because remember, the dominant overrides the recessive allele. All right, so now what does this mean for pedigrees? So this is a dominant pedigree. So first let's talk about what pedigrees are. So these are each generation. So generation 1, 2, 3. So if these are the kids, so this is like you guys, this would be your parents in generation 2, and this would be your grandparents in generation 3. All right, so the other thing you need to know is that circles are females. So all these people are females. And males are blockheads or squares. So all these guys are males. All right, so the last thing you need to know is the people who are colored in have the trait, and the people who are not colored in, no trait. All right, so now the reason why this is a dominant trait is because for everyone who you have all these, first of all, you have all these have it, and then for two people who don't have the trait, all their kids don't have it. All right, and so that's really only going to usually, I mean, it could happen if two people are dominant, but it only really happens if it's recessive, because remember, now you can only pass down a little letter to all your parents. All right, so that's usually the dominant, whereas these guys are probably, now this is the other reason you can tell is these two are obviously had to be dominant, but they had to pass down a little letter, so they both had to be heterozygous to give this person two little letters. And these guys are not halfway colored in; they're not carriers; they actually have the disease. All right. Whereas a carrier is someone who doesn't have the disease. So a carrier would be someone who doesn't have the disease but has the little letter, and what they look like is on the next slide. And this stuff you don't need to know too much. You're just going to need to know how to read the pedigrees. Those are going to be more important. Alright, so let's go to the next slide and look at a recessive pedigree. So now you can see there are people who are halfway colored in. These are all carriers. So since this is a recessive trait, the people colored in are recessive. So these guys are recessive here. So this would be little r, little r. These guys are big R, little r, meaning the reason why they're halfway colored in is because they carry the gene for that recessive trait, but they don't show it. They have the dominant trait. So let's say the big R is for round seeds and the little r is for wrinkled seeds. These guys, all the people halfway colored in, are going to be round or normal, like this says down here. All right. These guys, the ones that are colored in, will be wrinkled. So this guy is carrying the gene for the wrinkled wrinkled seed, but he would be round. All right, same with this guy, this guy, this guy, this guy, this guy. They're all carriers. They carry that gene for the wrinkled, whereas this one's going to be wrinkled. And then since this is dominant, these guys are not colored in. That means they are homozygous dominant. They're completely normal. They don't even carry that wrinkled gene. All right, so if you compare this to sickle cell, this would be someone with sickle cell. This would be someone who is carrying the sickle cell disease. So again, sickle cell is recessive, so this person doesn't have sickle cell, they're normal, but they're carrying the recessive disease gene. So what you're going to need to do is know how to read pedigrees, and then you're going to have to be able to make a pedigree, and that's the main activity, is to make a pedigree off of a gel electrophoresis of Anna's family. And you're going to make a pedigree of Anna's family and her inheritance. All right, so that is your goals for today. So again, once you take the pretest, you can skip the practice making and reading pedigrees, and you can go right to the activity.